Welcome back to a new episode of IBD Essential, a series established by Thomas Rösch and led by Axel Dignas. And we welcome today Dr. Remo Panaccione, one of the esteemed experts in clinical trials in inflammatory bowel diseases. And this series has been highly successful. But I want to point out you can also follow us on the channel. You can like us and you can comment on the videos if you have any ideas about how to make it better or things you liked and you didn't like in what we presented. So we're diving right in with Dr. Remo Panaccione from the University of Calgary. So Remo, welcome.
Thank you. Thank you for having me.
IBD Essentials. So let's start with how did you in fact end up in clinical trials? How did you end up in Calgary? You're leading a very established center there. How did all of this happen?
So I did my training at Western University in London, Ontario. And that's when I met Brian Fagan, who unfortunately has passed away recently. Good friend of both of ours. And so he introduced me to clinical trials. Then I did my IBD fellowship at the Mayo Clinic, where I was introduced to a young Bill Sanborn. In those days, we started doing some of the early trials in anti-TNF, and I got used to recruiting, and I was got interested in drug development. And then 27 years ago, I moved to Calgary. Lloyd Sutherland, who was a giant in the field at the time as well, he said, you should really build a clinical trial unit here. He was more into epidemiology and meta-analysis. And that's how it started. We started very, very small, and then we recruited other individuals. And so we've been fortunate that we have the patient population that engages in these clinical trials, is interested in participating. And so we've developed a successful program over the last quarter century.
Oh, impressive. So you learned from the best. And you're known to be an excellent recruiter for clinical trials and then obviously leading them. So perhaps we talk a bit about quote-unquote old drugs before we dive into the new mechanisms of actions. And what I want to start with is Vedolizumab. So Vedolizumab has been around for a long time, but recently there's new data emerging that helps us with positioning or with combination therapies. So can you guide us through LOVE-CD that recently was reported?
Yeah, I think, when we think about Vedolizumab, especially in Crohn's disease, it's been a little difficult to know where to position it. And I think, the two recent trials, both LOVE-CD, which was reported out last year and just published here in January, is really important because LOVE-CD was a trial that looked at intervening either early or later. So early was less than 24 months with no exposure to advanced therapies. Late was more than 24 months, patients on anti-TNF and immunomodulator. And what that trial definitively showed with a primary endpoint of clinical remission and endoscopic remission is both at the week 26 and week 52 endpoint. So over that entire year, that the patients that started early did much better. In fact, there was an 8-fold difference in the primary endpoint between starting Vedolizumab in that early population versus late. And it built on something else that we already know. There was less safety signals in the early patients versus the late patients, just reinforcing the importance of treating with highly effective therapy early on. And I think what it's taught us is also built on the post-op trial that was reported out last year as well, where it seems like if you take early disease or in patients who are post-op, Vedolizumab is a very, very good therapy. It may not be as effective if you're using it in later lines of therapy. So I think that will definitely change, or we should look at that space of where to introduce Vedolizumab in Crohn's disease.
So perhaps Vedolizumab so far underrated in fact for Crohn's disease, and this may give us a good clue about where to position.
Absolutely.
So about early disease versus late disease, there was a 2-year cutoff in LOVE-CD.
Yes.
Is that the right cutoff, or is early disease 1 year, 3 years, 5 years? How is that determined? What is your opinion?
Yeah, so I still think we don't know. Meaning, I think when LOVE-CD was designed and when it started to recruit, it was really based on those definitions that were based on expert consensus. 2 years, 5 years, longer than 5 years. And again, that was based on expert consensus, nothing else. I think we know from other studies that earlier is better and even less than 2 years. And I think we have examples like the profile trial that as early as you can start an effective therapy, it's better. So I would believe that even if you went to 1 year or below, the actual efficacy of any therapy, I think, will be better.
Better. Why do you think that is? So is that an immunophenotype that changes over time, makes it harder for biologics to work later on? Is it tissue damage? Is it both?
I think it's both. I think we know if you study patients, again, with early versus late disease, that the immunophenotype changes and that the pathways that are recruited that are driving inflammation will change over time. So there's probably a sweet spot early on. We also know that with inflammation, there's accumulated tissue damage. We don't have very good drugs that it can remodel tissue. So I think it's a combination. And in individual patients, what the driving force may be that makes them refractory or resistant to therapy may be very, very different.
Mm-hmm. Before we dive into combination with vedolizumab, is the early versus late concept also true in ulcerative colitis, or is this something that is particularly present in Crohn's disease?
As far as the observational data that we have, it's mostly in Crohn's disease versus ulcerative colitis. As you know, there was a meta-analysis done several years ago by Shamran that actually showed that, interestingly, that both the placebo rate and the drug rate in early disease with Crohn's disease was higher, but we didn't see that in ulcerative colitis. So the signal, for the reasons that we just talked about, is probably higher in Crohn's disease than ulcerative colitis.
I was a bit surprised about the Shamran result. I'm wondering if it has to do with the endpoint being clinical, and if you look deeper, endoscopic remission, histologic remission, molecular remission, if we find comparable phenomena also in ulcerative colitis, because it is a progressive disease too. You find fibrosis, you find other sequelae of chronic diseases.
Absolutely, I agree. And it gets to drug development, how we interact with regulatory authorities. I think that there's going to be ongoing in the next few years changes in how we measure the success of therapy. And I think that that's very good, not only for drug development, but I think that it will make treating in the clinic probably easier.
Mm-hmm. So you mentioned safety earlier in LAUF-CD, and vedolizumab in general is considered a very safe choice for our patients. And hence, with emerging combination trials, it is a very sought-after combination partner. You are heavily involved in the Explorer combination trial with vedolizumab. Can you talk us through which population was that tested in, which combinations, and then what was the outcome?
Yeah, so I think that there was when Explorer was designed, it really was trying to think about the future of where we're going to go in inflammatory bowel disease. And I think you've outlined nicely the fact that we do think that for many reasons that we probably need to use combination therapy. And vedolizumab is a natural foundation for that. So in the Explorer study, it was a high-risk patient population, which again was on expert consensus. And it used a combination of vedolizumab, methotrexate, and adalimumab. And it was performed over a period of 40 weeks. And the high-risk patients all got the three drugs together. And then the adalimumab was taken off after 6 months, and then the methotrexate after 9 months. And the primary endpoint was clinical and endoscopic remission. There was no placebo group in this. The placebo was a Bayesian placebo, was modeled. And it showed that, that it was about 35 to 40% more effective than what you would get with placebo. And there's a great interest now in combination with vedolizumab. So that was Explorer 1.0, a larger study. Explorer 2.0 is just being launched now. And there's combinations with vedolizumab with some of our, with JAK inhibitors. So there's a vedolizumab and tofacitinib trial in ulcerative colitis. There's a vedolizumab and upadacitinib trial in Crohn's disease, trying to boost, the induction phase of our treatment regimens and see if vedolizumab, once you get patients in remission, may be similar to what we think about post-op, where if you can actually get the patients in a good state, that vedolizumab is a very good and safe maintenance therapy.
Mm-hmm. And thank you for contributing so much to this area. And one of the abstracts here at ECHO, in fact, we are recording this, is about a combination therapy of vedolizumab and JAK inhibitor coming from China. Very impressive recruitment rates and now very high quality data coming from there also.
Yeah, in that group, presented some early preliminary data, I think a year ago. And I think we're very enthusiastic about combinations, especially when you can have a very safe partner with it.
Mm-hmm. A different safe partner would be anti-IL23 medications. This is a crowded field now, so we have 3 approved anti-P19 drugs: risankizumab, birinkizumab, guselkumab. Can you lead us through the clinical development programs, how these drugs, which populations they were developed for, and how do you use them in practice?
Yeah, I think that in general, I'll start off saying that I think anti-P19 therapy is a game changer. Regardless of the drug that we're talking about, as far as the 3 that are on the market, I think namely because you get very, very high efficacy across a lot of endpoints, and you're not really seeing a safety signal. And we know that from the development in psoriasis. So when you think about, the balance between efficacy and safety, it's hard to, conceive at this moment a class of drugs that balances that more than the anti-P19s. So, perhaps we'll start with risankizumab. Risankizumab was developed, in a very robust phase 2 program in Crohn's disease and ulcerative colitis. And I think the key to the risankizumab development is the way they selected doses during induction. I think their phase 2 program was done very well, which has led to different doses in moderate to severe Crohn's disease and ulcerative colitis as far as the induction doses. So 600 milligrams in Crohn's disease, 1,200 milligrams in ulcerative colitis, followed by the subcutaneous maintenance. Met all the primary and secondary endpoints, obviously a drug that we're now using in clinical practice quite often. Moving to guselkumab, I think that program was a bit different. Started with Galaxy 1 in Crohn's disease, and then we have the Galaxy 2, 3 studies in Quasar and moderate to severe ulcerative colitis as well as Astro and Galaxy checking sub-Q induction. So I think the hallmark of that drug is its flexibility during induction, because all of those studies showed efficacy during induction. So you can do IV induction or sub-Q induction. There's really no difference in those patient populations. I mean, in the efficacy between sub-Q and IV. And then you can move into maintenance with either a 100 milligram dose every 8 weeks or 200 every 4 weeks. The one thing that's important, I think, which is a little confusing because you have 2 maintenance doses for that drug, is to understand which patients do better on the 200 versus the 100. Because if you look at the efficacy in the primary results, they look almost the same, right? But there are subpopulations that do better with the 200. Patients with sort of more severe disease up front, patients who don't respond after induction, high CRP, high fecal calprotectin, or previous drug exposure probably do better with the 200. There's a lot of talk about the difference, in target engagement, because guselkumab is a native FC, so interacts with CD64. I don't know what your opinion is, but, we have a lot of in vitro data. I don't know if it adds to the efficacy or is going to add to the durability, but that's something the audience will hear about from others, I would say. And then we have mirikizumab, which has, good data in moderate to severe ulcerative colitis and Crohn's disease, the first anti-P19 and ulcerative colitis. The difference about that development program is similar to the Galaxy program. There's a comparator against ustekinumab, and unfortunately, for probably many reasons, it wasn't superior to ustekinumab as part of even for the objective endpoints as opposed to guselkumab. So I think in clinical practice, you really have, guselkumab and risankizumab look very similar. I think mirikizumab, the data, just if you look from a pure efficacy, not an absolute efficacy, but the comparator, it was a negative study for some of the endpoints. But, for our colleagues here in Europe, I think it's a more cost-effective option, I think, in many countries. And I don't think you can go wrong with either one of them.
My opinion, so I agree with everything you said. I think the three molecules in my book are fairly equivalent. I think there were potential reasons why mirikizumab performed less well against ustekinumab compared to the other trials. And there's a big battle for differentiation, and you already hinted towards CD64. The way the CD64 experiments were done, perhaps not under physiological conditions, because in the serum or in the tissue, CD64 in general, it's maybe too much detail, is saturated likely already by native IgG. I'm not sure how much the binding of extra antibody makes a difference, but the clinical efficacy of all three drugs is very good. It's very safe drugs, and at least in the US market, they are very strong for good reason.
And, and to build a little bit on that CD64, we know that, for example, ustekinumab binds CD64, and when you look at the comparison, either in Galaxy or if we didn't talk about Sequence, which with risankizumab, it didn't seem like it boosted the efficacy.
Yeah. Ultimately, clinical efficacy, I think, will rule.
Yeah, I absolutely agree.
So let's talk about speed of onset and durability. So there's a lot of work being done on how fast the drug works, how fast the patient feels better. As an example, anti-TNF patients receive infusion. Some of them say even within a day they feel better. JAK inhibitors show clinical improvement within a few days. How do the anti-P19s fare in that space? And then once you're in remission, once you have mucosal healing, what is the durability?
Yeah, so, and we've seen some data, there's going to be more data here as far as the long-term extension studies. Anti-P19s, I think, are relatively fast. We think about anti-TNF and JAK inhibitors as probably our quickest therapies in getting patients better. But I always say that there's a caveat when we talk to patients about that. Yes, they work quickly, but they don't work quickly in everyone. So, in clinical practice, even when I use an anti-TNF or a JAK inhibitor, I said, you could get better, as soon as in days or the first couple of weeks, but some patients take longer. And I just wanted to mention that because I think it's important, because some individuals, if they don't get that wow effect with an anti-TNF or a JAK, they think the patient may not be responding, and they may make, premature decisions, as I call them. Anti-P19s, usually we can see in all of the trials within the first dosing, IV and or sub-Q, within the first 2 weeks, you usually have at least a clinical response in 30 to 40% of patients. And then certainly after the second dose during induction, you capture most of the patients. There is, again, a population of about 30% of patients that may need extended induction. We just published on that just recently. But I usually tell my patients, within 2 weeks, you should notice at least a response in your symptoms.
And do all anti-P19s allow extended induction and then reinduction later on?
Yeah, so this is now a very muddled, and how we name it, how we call this. So just because of the way the trials were designed, so the trials with guselkumab, at least the Crohn's disease trials, and mirikizumab were treat through designs, right? So there was escape arms, but there was no extended induction in those trials. The only data that we have is with risankizumab in Crohn's disease, where patients could either receive more IV if they were non-responders, or transition into the doses that were used during maintenance. And in those trials, what we saw in between week 12 and 24 is about 60% of the non-responders responded, and about a third of the patients went into remission. But you didn't need more IV, you just needed more time with it on drug. Mirikizumab in ulcerative colitis has an extra dose, and I think in the United States and other places you can use that. So they have that data. We don't have that data with guselkumab. We really don't know what that rescue is. The only data we have in dose flexibility there is at the end of the 48-week trial, and I'll be presenting some of that at this meeting in DDW.
Mm-hmm. 2 questions before we wrap up, because we're already coming towards the end of time of this podcast. Durability. So patients that are in remission after induction, in remission after maintenance, how do they fare over the long run?
Yeah, I think the durability, if we're sticking with P19s, is quite good. I think we saw that in psoriasis. The durability in psoriasis is very good. And we're seeing about a 10 to 15% delta over what we would historically see with anti-TNFs. Now, is that because of efficacy? Is that because of lack of immunogenicity with some of the newer antibodies? I think we really don't know. But I think that once patients get well, they seem to stay well. We're going to see data here with mirikizumab almost out to 4 years, 3 years with risankizumab, 2 years plus with and it all looks pretty stable. So the durability seems to be quite good.
Comparable to vedolizumab. Vedolizumab is a very durable drug after your remission.
Yeah. So I think once you pick the winners, and when we think about durability, we think not only about efficacy, but there's probably a safety aspect to that, right? So it's more or less persistency, and because these drugs are safe, I think we're seeing patients stay on them long term.
Last question before we wrap up.
Yeah.
Patient on ustekinumab has ulcers on endoscopy. Can you switch them to a P19? And if so, which one? And what's the evidence?
So it's a great question. And so there's two sources of evidence. So in the risankizumab trials, almost 20% of the population was exposed to ustekinumab. And those patients had to have active disease endoscopically, which was centrally read. And the endoscopic response rates and remission rates, albeit absolutely lower, the delta between placebo is like being exposed to other therapies. More interesting, and what the audience, if they're listening to it after ECHO, there's a very, very nice study that just is going to be presented here at ECHO from Leuven, Belgium, where they took patients specifically who were in symptomatic remission but had endoscopic activity and switched them to risankizumab. And in that study, they actually saw that a fair proportion of patients had an endoscopic response. I think just showing us that selective sort of IL-23 inhibition does have an advantage, even objectively, over anti-IL-23.
So a true treat-to-target approach then. So patients without symptoms but endoscopic lesions, and you switch therapy based on endoscopy only, and improve outcomes.
Yeah, and I think that's so very important, because I think in some people in clinical practice are a little hesitant to do that when the patient feels well. So I think that, even though not sort of a phase 3 or even a phase 4 trial, very, very important data.
So thank you very much. This was excellent. An excellent update on vedolizumab and anti-P19. So much more to talk about. We will definitely have you back in the future, talk about novel therapies, anti-TL1A and so forth. We appreciate a lot your time and your expertise, and congratulations on a very distinguished career.
Oh, thank you very much, Florian, for having me, and thanks for the audience for listening.
So thank you for tuning in today to IBD Essentials. Remember, you can like us, and we would really like to hear your comments once you follow our channel. So thank you, and hope to see you next time.